Hi everyone, my name is Maya Groff and I'm a postbaccalaureate fellow in the Con Lab at the NCI, and I'm so honored to be here today to discuss with you all our research looking into how developmental states determine intrinsic resistance to immunotherapy in the pediatric cancer rhabdomyosarcoma. So rhabdomyosarcoma, or RMS, is the most common soft tissue cancer in pediatrics. It has a 5-year overall survival of 85% in localized disease, but only 17% and 30% in recurrent and metastatic disease, respectively. It's classified into two major subtypes, fusion negative and fusion positive, named based off of the absence or presence of a transcription factor fusion between the genes PAX3 and 7 and FOXO1. In general, fusion-positive RMS is associated with more aggressive presentations and poorer outcomes. The current standard of care is chemotherapy followed by radiation and surgery, and this results in cure for about 90% of patients with low-risk disease, but this treatment is very cytotoxic and leaves children with side effects that follow them throughout the rest of their lives. Survival also remains low in high-risk progressed disease, with an event-free survival of around 20%. And there's been no improvement in clinical treatment for these patients in the past 30 years, highlighting the very urgent need for new therapies being developed. Attention has turned to various types of immunotherapies like monoclonal antibodies, checkpoint inhibitors, adoptive cell therapies, cancer vaccines, and oncolytic viruses. And there have been clinical trials exploring therapies like peptide vaccines and stem cell transplants for the treatment of RMS specifically. And though immunotherapies in general show promise, with the state of the field to date, there is still significant progress that needs to be made. Our lab focuses on exploring the immune landscape of pediatric cancers in order to improve and enhance different immune therapies. Immune therapies can be broadly distinguished based on if they target surface proteins or if they target antigens presented by major histocompatibility complex class 1 or MHC class 1. Surface proteins are mostly targeted using antibody-based therapies, including CAR T cells, which we've heard a lot about today. Our lab will be opening a phase 1 clinical trial at the NIH for a CAR T therapy targeting FGFR4 and RMS this summer. The other class of targets for immune therapies are MHC1 presented antigens, and these include things like neoantigens, fusion breakpoints, and cancer germline antigens. These can be targeted with endogenous or engineered T-cell receptors, and our focus that I'll continue to discuss throughout this presentation is on engineered T-cell receptors, or TCRs. TCRs depend on the expression of antigens on the surface of cells by MHC1. So in this process, the antigen processing and presentation pathway, large proteins are broken into smaller peptides, which are picked up by MHC1 and then expressed on the surface of cells. TCRs are then able to recognize the antigen presented by MHC1, and this initiates an immune response against the tumors. MHC expression in different tumors is very dynamic, with some having high expression while others have very low expression. And it's known that downregulation of MHC1 is a mechanism of immune escape used in tumors. And this low or suppressed expression of MHC can pose a serious limitation to the use of immune therapies, including those like TCRs. Understanding this challenge, our lab dug deeper into the expression of MHC1 in a variety of different pediatric cancers, exploring its expression on the surface of cells. We included controls shown in gray, which offer an idea of what a spectrum of MHC expression looks like. So this includes human fibroblasts, which have high or normal MHC expression, a breast cancer cell line that's known to have low expression, as well as a gold standard of almost entirely absent expression. And we show here that pediatric cancers have a range of MHC expression, with some like osteosarcoma having high expression, while others like MYC amplified neuroblastoma have very low expression. And then looking specifically at rhabdomyosarcoma, we saw that fusion-negative RMS has a spectrum of expression, while fusion-positive RMS has entirely absent or very low expression of RMS. And we confirmed this by RNA sequencing, examining the expression of one of the genes that encodes for MHC1, called HLA-A, comparing adult tumors to pediatric ones. And when we did this, we saw that in general, pediatric tumors have low expression compared to adult, and specifically this is seen in RMS. There have been several studies investigating what might be causing this loss, and there's been no findings that genetic mutation are the cause of this suppressed antigen presentation. So this prompted us to explore what might be causing the low expression of MHC in RMS. In this project, we went about exploring the etiology of the differences in antigen presentation, and then identified pharmacological agents that restore antigen presentation and synergize with TCR immunotherapy. We hypothesized that the expression of the antigen processing pathway is associated with a myoblast cell state mimicking normal embryonic myogenesis. In normal muscle development, progenitor cells evolve all the way into fully developed myotubules, expressing different transcription factors at each stage. So progenitor cells evolve into satellite cells that express PAX7, then into myoblasts that express MyoD, then into myocytes expressing MyoG, and finally into fully developed myotubes. RMS mimics these different stages of development, with fusion-negative RMS resembling a spectrum of stages from the earliest of progenitor cells to fully developed myotubes, while fusion-positive RMS mostly mimics a myoblast myocyte state. And we see these trends reflected in the immunohistochemistry on the right, where we see that fusion-negative RMS expresses the transcription factors PAX7 and MyoG, revealing a spectrum of developmental stages, while fusion-positive RMS expresses strictly MyoG, highlighting that it exists mostly in a myoblast myocyte cell state. Knowing that RMS mimics these different stages of development, we explored how antigen presentation is affected in each of these stages in normal muscle development. So we used a stem cell model that mimics the different stages of development created by this lab group here, where they differentiated stem cells into each of the different stages of development, and then selected for the cells in each of these stages using a GFP reporter that followed the transcription factors predominantly expressed in each of those stages. And then we were able to use those pools of cells as a model. And we used them to look at the RNA expression of genes involved in antigen processing and presentation, and saw that in early and late stages of development, there's very high expression of the genes involved in antigen presentation, but in myogenic states, there's very low expression of the genes involved in antigen presentation. So establishing that in normal muscle development, each stage has different levels of antigen presentation, and knowing that RMS mimics these different stages of development, we wanted to explore if there was a relationship between embryonic myogenesis and antigen presentation in RMS. And we did this by creating a gene signature of the top 100 genes that were elevated and diminished in each of the developmental states of normal muscle development compared to all of the other states, and then conducted gene set enrichment analysis and correlated the gene sets for antigen presentation and myogenic states in these RMS tumors. When we did this, we saw that the genes expressed in early presomite stages as well as in late stages of myotubes, there's positive correlation with the expression of genes involved in antigen presentation. On the other hand, when we're looking at genes that are anti-correlated with a myogenic myocyte state, we see very strong positive correlation with antigen presentation. So to summarize these findings clearly, we demonstrated that genes expressed in the early and late stages of development are very positively correlated with antigen presentation, but in myogenic states, there's very low expression of the genes involved in antigen presentation. And we validated this on a protein level, looking in a variety of fusion-negative and fusion-positive patient-derived xenografts. And we saw that fusion-negative RMS tumors express a range of proteins involved in antigen presentation, while fusion-positive RMS expresses virtually none of these proteins. And then when we probed for myogenic markers MyoD1 and MyoG, we saw that tumors with high expression of the antigen presentation proteins had no expression of MyoD and MyoG, whereas tumors that had low APP had very high expression of these markers. And so this demonstrated very clearly that the myoblast state is inversely correlated with expression of antigen presentation. So understanding all of this information and seeing the relationship between developmental states and antigen presentation in RMS, we wanted to explore if there were drugs that could be used to boost MHC expression in these tumors. So there have been other studies that show that certain epigenetic drugs can effectively boost antigen presentation, and these drugs work by increasing overall transcription, increasing antigen expression, tumor antigens, and endogenous retroviruses, and we wanted to explore if they could be used to enhance APP and RMS and sensitize tumors to immunotherapies. We treated a panel of RMS cell lines with the epigenetic drugs decitabine, mosatinostat, and tazemetostat for 5 days, and saw that some of these cell lines shown on the left respond extremely well to the drugs, specifically to decitabine and mosatinostat, while other of these cell lines shown on the right had modest or no responses. When we added interferon gamma, which is a cytokine that stimulates an immune response, we saw that cells that didn't have a significant response to the drugs alone continued to have little response to interferon. But when we combined interferon with these drugs, we see synergy between the two agents and increases in MHC1 expression across all of these different cell lines. So because of this finding, we wanted to explore if we could use these drugs to enhance responsiveness to immunotherapies and RMS. And to investigate this, we used a TCR immunotherapy that targets the antigen PRAME. PRAME is a cancer germline antigen that's highly expressed in a variety of different extracranial solid tumors, including RMS, with little expression in normal tissue except for in the testes and ovaries, making it a very valuable target for TCRs. We treated a fusion-negative RMS PDX that's positive for PRAME expression with decitabine for 5 days to sensitize the cells, which we validated by increased expression of antigen presentation proteins, as well as increased expression of MHC on the surface of cells. And then we targeted the tumors with the TCR and saw that treating the cells with decitabine significantly increased T-cell mediated killing and improved the effectiveness of the PRAME TCR. So this study very clearly demonstrated that the epigenetic drugs can sensitize RMS to immunotherapy and make it more effective. So in this project, we were able to show that pediatric RMS has a deficiency of antigen presentation, and that deficiency is associated with a myoblast myocyte cell state. And then we were able to demonstrate the ability to reverse low antigen presentation with epigenetic drugs, which ultimately increase the sensitivity to T-cell receptor therapies using PRAME, targeting PRAME. Moving forward with this project, we'll begin in vivo experiments looking at the combination of epigenetic drugs and PRAME TCR in targeting RMS. I'd like to offer a big thank you to Dr. Javed Khan and Dr. David Maluski, who were the minds and energy behind this project, as well as the rest of the Khan Lab for all of their support. And thank you all for listening.
Hi, thank you. Such an interesting talk. My understanding of these HDAC inhibitors is that they're opening up transcription pretty generally and not necessarily in a targeted manner. So it's fortunate, it's really cool you prove that it results in a direct increase in the MHC presentation, MHC transcription. Are you concerned at all about off-target or other ways that it will open up transcription and cause other things that may complicate things or cause unintended effects?
Yeah, definitely. That's a great question and a possibility. So as I mentioned, these drugs work through a variety of mechanisms and, as you said, increase transcription overall. So we were focusing specifically on the expression of MHC in tumors, but it is a possibility that the drugs have an effect on other things going on within the tumor T-cell environment. And so one thing that's been seen is that they can actually have an effect on T-cells. When we've done our assays treating tumors with drugs, we pre-treated the tumors, so it's focusing just on that effect. But when we begin studies looking at in vivo experiments, we'll do drug and T-cells together, and we'll be able to take a look at what sort of effect these drugs might be having on the T-cells and sort of get at what you're asking and see if there might be other effects going on.
Beautiful talk, Maya. Thank you so much. I had a question about how FOXO fusion status may or may not regulate antigen presentation and some of the effects that you're seeing. I thought perhaps in one of your figures you showed the cells were more or less segregating by fusion negative and fusion positive in their expression of antigen and then their expression of MyoG. But when you looked at the epigenetic modifying drugs, sensitivity, according to the cell lines that you showed, didn't seem to follow that same pattern between fusion negative and fusion positive. So I'm wondering, to what extent is FOXO regulating this or is not?
That's a great question. Yeah, so in general, fusion positive RMS is associated with lower MHC expression, and we showed that through the flow cytometry, seeing the changes in MHC expression. But like you said, the response to the drugs was scattered between the two groups in terms of which were more effective and which were not. So that's definitely something that we could continue to look into as we move forward, exploring if the FOXO1 fusion has an impact on MHC expression. Awesome. Thank you.